just to acknowledge the people that helped make this possible. And thank you to everyone who's here right now. I just want to also let you all know that this session will be recorded because since this is a global event, some people are right now. So a lot of people are watching the recorded video after. I'm going to hand this over to our moderator, Dr. Eric McLemore. Thank you so much for joining us today, Eric, and to our speakers, Adrian and Brendan. I will let you take it from here. Thank you.
Thanks, Chelsea. Thanks, everyone, for joining us. We're very excited to kick off our Oceania session here in GARD. Welcome to those of you on the East Coast in the US. It's late in the evening. Welcome to everyone who's on a different time zone. Our first very exciting speaker is Dr. Adrian Dinsdale from the Innovation in Plant, speaking in the topic of innovation in plant biosecurity from concept to implementation. Dr. Dinsdale is the acting director of the Plant Innovation Center and devotes energies towards identifying and developing new technologies, improving operational outcomes, building relationships with external scientific agencies, and driving education sector engagement. If you take a look at the profile in Huva, he has a pretty extensive background in and out of academia and industry and some areas in between. So please join me in welcoming our speaker, Dr. Dinsdale.
Thanks very much, Eric. Now, forgive me, I'm a bit of a novice with Zoom, so I'll try and share my screen. While I'm doing that, I'll make some idle chit chat and thank you very much for the invitation. And it's particularly a pleasure to be at one of these events stateside where it's within my normal working hours. Normally I'm up very early for these things, so thank you very much for that. Now, I think I'm sharing. Is that working? Yeah, okay, you can see my slides. All right. Okay, great. I'll jump into it. So as Eric mentioned, I work for, sorry, hang on, I can't see my slides now. That's better. I work for Australian federal government, Department of Agriculture, Fisheries and Forestry. So one of our many areas of remit is border biosecurity. And that's sort of mostly where my talk's going to talk about today. Now, forgive me if I'm telling some of you how to suck eggs, but I do like to kind of start these talks right at the very kind of broad, higher level concept of why, why we even do the work we do, why is it important? And for me, it really comes down to sort of 3 or 4 key concepts, and essentially they can be summarized as food, money / jobs, kind of the same thing in some ways, and environment. So Australia is a really massive food producer and exporter. As you can see there, it creates a lot of jobs, creates a lot of income for our country, and really importantly, it feeds a lot of people. So it's really critical. But also, as you probably know, Australia is a pretty unique island continent. And therefore, as you'd expect, a lot of our flora and fauna are quite unique. And so that needs to be protected as well for a whole lot of obvious reasons that I'm sure I don't need to go into here. So that's what we're trying to protect, but it's getting harder and harder to effectively protect our border from exotic pests and disease. So the slide in orange there, I'll see if I can get my pointer to work. There's the pointer, there we go. Hopefully you can all see that. The slide, the image there at the top in the orange, those figures are a little bit dated, but that's just a quick snapshot of the volume of stuff that enters Australia annually and comes across our border. And every one of those represents a potential biosecurity risk, and some of them are an actual risk. But because every one of those items has the potential to be risk, every single item has to be somehow managed to mitigate that risk down to a low or preferably zero level. The scary bit though really is that image there in blue underneath shows the projected increases. That's going to really put a massive strain on our ability to keep doing what we do effectively. So some modelling has been done internally. I think it's internal, maybe it's been external, and that really hit home the point that we just can't keep doing the business that we do in the way we currently do it and manage that risk. So I think the modelling showed that even if we do, I think it was almost a threefold increase in the current funding that we have to manage border biosecurity, it was still projected to at best just keep up with that increase in movement of goods. So we're not going to get a threefold increase in our funding in the next 2 or 3 years. So that's not sustainable. We have to find ways to do what we do better. And I'll come into that. I'll go into that a bit more detail shortly. Not surprisingly, those increased movements of goods turn into increased rates of detections of biosecurity risks and so on. So look, these figures, the details don't really matter. All that really does matter is that you can see that the risk is growing and the rate of interception of biosecurity risk is growing. And indeed, the cost and the effort and the resources needed to continue to maintain the ongoing protection and the response to those detections is growing. And again, that's putting massive pressure on not just our operators at the border, but also our colleagues in the state government who have to effectively carry the can when things do get over the border. So that's in a real nutshell what I hope you can get from that. Why we do biosecurity, why it's important to do it, the current state of where it's at, where it's projected to be at, and hopefully what all that will bring home is that we need to do biosecurity smarter, faster, more efficient, etc. So, i.e., we need to innovate, and so that's where the team that I have the pleasure of leading comes into play. Our team's called the Plant Innovation Centre, and we're the department's dedicated, it's a bit annoying, dedicated plant biosecurity R&D team. There's a little QR code there if you'd like to learn a little bit more, or you're interested in student opportunities. It's still a pretty new website. It's a work in progress, but it is there, and it's an interesting kind of first stop, find a little bit more about some of the work we do. So we are based down here in Melbourne. So Melbourne is in the very sort of southeast corner of mainland Australia. This entire site is not ours, sadly. It's a shared site. But what this actually is Australia's national high-risk, pardon me, post-entry quarantine facility. So almost all high-risk imported, living things that come into Australia must come to this facility. There's always the odd exception, but by and large, that's the general rule. So it's a big site. It's about 300 acres all up. Opened quite recently, just shy of about a $500 million price tag. So, I think this really kind of, again, embodies in some way our government's commitment to biosecurity and trying to keep a lot of those exotic pests and diseases out of Australia that we're free of. I'll give you a super quick little virtual tour of the facility. So here we have, this is our cat facility, holds about 250 odd cats. This big one here is full of dogs, holds about 400 dogs. This little guy at the back here is our bee facility. This one here is for ruminants. This big one here is for birds and eggs. Here we have horses, and another horses, and then lastly, there is the plant facility, which is where, of course, our team is based. Nice little aerial shot there of the plant facility. These are all of our, whoops, there we go. These are all of our high containment biosecurity glass houses. So most of the material in those glass houses is all what we would call sort of high risk nursery stock material. So imported plants, new genetics that is going to go into primarily agricultural either breeding programs or trial programs to try out new cultivars, to see how they perform in Australian conditions. And this long skinny building here that's connected to all those glass houses is our laboratory complex. It's worth noting that, and forgive me for any animal colleagues who are present, that the plant facility is the only part of our compound or our site that has a dedicated laboratory facility and teams of scientists who are employed there full time. So we have a team of full-time diagnosticians. It's their jobs to test and screen every single plant that comes into quarantine to make sure they're free of any biosecurity risk before being released. And we have an R&D team based there as well. And in a nutshell, because plants are really hard, and sorry that animals, from our perspective, they're really easy. We can have 700, 800 cats and dogs and horses on site, and even though they come in all different shapes and sizes, ultimately we're still looking at 4 different species. That's it. And each one of those species is really quite well characterized and understood. We really have a pretty good handle on all of the pathogens and the host relationships with those pathogens, how they're vectored, how they're detected, how they're treated, and so on. With plants, we don't have that luxury. At any given time in this facility, we will have 300 to 400 plants, so that will be across, on average, 20 to 30 genera. Multiply that by a factor of X for species, and that gives you a sense of the complexity of what we have to deal with from a diagnostic perspective. Add to that, well, dogs and cats or animals have quite short stays in quarantine. So animals, average sort of 2 to 3 weeks, depending on the animal and where they're coming from. So for a cattle dog from the US, you're looking at 30 days because they're in this country. Otherwise, it's only 10 days. That's it. You're in and out. With a plant, we're looking at an average of a year, up to 2 years. If you're lucky, maybe 6 months. So just way more complicated, harder to manage. And I'll just quickly here mention what I always mention on these talks when I have an international audience. If you're considering coming to Australia, I don't recommend you bring your pet cat or dog, whatever. It's really expensive. It takes a long time. It's really complicated. We don't make it easy. We provide, a first-class service, and we'll absolutely manage the hell out of that risk. But you're not going to get any change out of, $5,000 to $10,000, assuming it all goes kind of reasonably well. We have had some horror stories where very occasionally an animal has had to overstay. I think the record I know of is 3 months. That's a long time, and it's going to certainly stuff up your holiday plans. And really sadly, on occasion, we sometimes have to destroy an animal. Sometimes they come out of the cargo hold of the plane already dead. It's really stressful on your dog or cat. So that's just my little plug to really not do that lightly and think long and hard about that decision. Okay, so that's the team, and that's where we're based. And I hope that gives you a little idea of why our team exists to do the work we do. And now I'm going to talk a little bit more about some of the actual innovation work we do. A lot of this is based on diagnostics, but not entirely. Sorry, pardon me. So these are some of the projects that we're currently working on. This is not exhaustive. I've just picked a few out that I think are pretty interesting and hopefully relevant to this audience. So the first one is not diagnostics, but it's worth mentioning So MBR, that's methyl bromide. It's a really, really super effective fumigant, and for us, it's a fabulous tool in our toolkit. It's super effective. It kills everything, but it doesn't damage the things you're treating too much. It kind of dissipates and breaks down really quickly, so it's reasonably okay to manage the health and safety risk, and it penetrates quite well into things as well. So this is almost our go-to. So when we have goods detected at the border and we find a problem and we're not sure what to do about it, this is the go-to. Okay, methyl bromide, bang, risk managed. That's all fine, but methyl bromide is also a super nasty ozone depleter. And in fact, it's been banned worldwide under what's called the Montreal Protocol. In the EU, it's completely banned outright, but in most other countries, there is a little clause in there that says you can still use it for quarantine applications. So Australia has really, unfortunately, become a bit of a pariah with our use of this fumigant. We use it a lot. We probably use it too much. So we really need to develop alternatives to this fumigant and pretty quickly. So we're working with a couple of project partners developing methyl iodide as an alternative, and also playing around with microwaves, which look like they'll be quite effective, but for a smaller subset of things to treat with it. Moving into diagnostics, we are working increasingly with CRISPR. I think this is something this is going to be, the future of diagnostics. Just its power of specificity and great sensitivity give this lots of applications for us that I think is really quite interesting and exciting. What we really are hoping to develop is something that is effectively, a lab on a chip. What I'd love to see is for us to be able to do away with a lot of our labs and instead equip our officers in the field with these really powerful portable devices to allow them to make triage decisions in the field with confidence. And I think CRISPR could be part of the answer to that. I'll talk more a bit about that in a second, but we've done quite a lot of work with that, a lot of proof of concept work that's been successful and been published, and it's an area of growing interest for us. We're also doing a lot of work with honeybees. So Australia was the only honey producing continent, so everywhere except Antarctica, that was free of what I think is pretty well accepted as the world's worst pest of honeybees, which is something called Varroa destructor, that little red blob there on the side of that unfortunate bee, the little mite. We are in the midst of an eradication response at the moment, so technically at the moment we're not free, but we're having a bloody good crack at trying to eradicate that. Our current testing is not very great, so we're doing a lot of work with CSIRO, which is the Commonwealth Scientific and Industrial Research Organisation, a government-funded group, to develop new molecular diagnostics to detect these mites. So what we want is one test that will detect all exotic mites in one go rather than separate tests, which is what we do at the moment. This project at the top there, nanoparticles, we're exploring a few different technical platforms for this one, but again, one of them is CRISPR. It's not limited to CRISPR, but essentially, as I kind of already touched on, this is the next stage of some of that work. So we've done the work in our lab to show the concept that the CRISPR can be used to detect a range of important plant pathogens that we're concerned about. Now we're working with this nano biosensor group, RMIT University, who have these nano fabrication capability to start to actually turn that CRISPR work into a physical kind of prototype that we hope to be able to one day make field deployable. What I think is particularly neat about this one is that we are working in parallel with our second project in microneedles with the University of Southern Queensland. So there's a nano microfabrication engineering place That was eloquent, wasn't it? At this university. And we're working with them to play around with a whole bunch of different polymers in these microneedles, which are, as the name implies, as simple as they look. They're quite simply very tiny microneedles. You know, the real problem with doing this work on plants is if you just take a chunk of leaf tissue or whatever and you throw it in the tube and you just grind it up, there's all these really crappy, you know, complex polysaccharides and stuff in there that really wreak havoc with your downstream applications. You can't get sensitive diagnostics with that crap still in there. And so you have to go through a pretty complicated and can be unpleasant process to purify that material. So the idea with the microneedles is there's that, they quite simply, you puncture your tissue with these microneedles. Their structure has built-in microfluidics to draw in material. They are made of a polymer that's highly hydrophilic. And so again, it draws in all the fluid. So you pull it out, and then what you instantly have from your microneedle is a purified template for downstream application. So again, we've done this work in our lab and showed that it works quite effectively to be able to detect infections in plant hosts without any additional work needed. So the dream is that we'll use our CRISPR technology deployed in a nanobiosensor attached to a microneedle to give us an all-in-one field deployable lab on a chip. We're still some years away, but you know, we're making kind of steady progress towards that. And the last project, just thought I'd mention that we're currently working on, thought I'd throw it in there because it's got some a US connection, working with the University of Florida. And effectively what we're trying to do is do away with the amplification step when we're trying to detect pests in imported seeds. So rather than making amplicons, we are generating a whole bunch of these tiny little probes or tiles that go across the genomes of this, in this instance, viruses of concern. So you do your extraction, essentially apply your probes, and then you run it through a MinION. All right, so that's just some of the work that we have underway. That's a bit of a cross section of the kind of breadth and depth, I hope, of our work. This is a few of our innovations that have actually been implemented. I wish there was more of them, or well, there is more of them. I haven't got them all here. Some of them are kind of boring. But we're still a relatively new team. Our group only came into being in 2017. So I think, you know, we've done pretty well to get the runs on the board that we have. The first one is, again, utilizing MinION, which is just a really great, super powerful technology. Big issue for us, being a border regulator, is that, you know, is time. So the place I talked about, the quarantine facility, like I said, you know, plants locked up there for months, years. So we have the luxury of time. We're the only laboratory in the department that has that luxury. All of our other labs are based around the points of entry into Australia, so major ports and airports. And that's where most of the goods come in. So if you are presented with, you know, a container load of whatever, something perishable, Brussels sprouts, and you find some insects in there, let's say they are, you know, larvae or something, and you just can't get a confident ID on that from morphology, we can't wait a week to get a Sanger sequencing result on that to confirm the ID, particularly if it's in the middle of summer. So we've worked up some really nice, rapid, robust barcoding methods with the MinION to be able to turn that around now in just a few hours. Cut flower devitalization, most people are pretty surprised to learn that every single cut flower that comes into Australia is actually poisoned with Roundup or glyphosate. That's a mandatory treatment. The problem for us was that we were never really 100% sure if the flowers coming in were actually treated the way they were meant to be. So we worked up a new method using HPLC mass spec to not only quantify that the treatment had been done, but also measure some degradation products of the glyphosate to put some quality controls in there as well. And just lastly, E-beam, so that's electron beam, it's a radiation. We've been able to show that we can use that to really quickly and safely and effectively treat imported grains and devitalize them, which i.e. means that they can't germinate. That's really handy for us because if you're importing these seeds for consumption, we don't want to put you through a full regulatory, rigor to manage that, because when you eat them, risk managed. But at the same time, we want to be sure that if you import them for eating, you can't just then divert them and put them in a field and completely circumvent that biosecurity regulatory net. So we look at these kinds of treatments. What we are always looking for: quick, cheap, effective, to again manage that risk without putting, undue impost on industry and importers. Now I'm going to spend a little bit more time delving into a bit of a case study. This one's not a rapid diagnostic, so forgive me if I'm self-indulging, but it's a really nice little story of an implementation, again, of technology, of innovation into a really highly regulated setting. So as I mentioned, at the quarantine facility, that's where we lock stuff up and make sure it's safe before we release it. And we do that with a whole mishmash of all kinds of different tests. And I've just listed some of them there under the methods. I won't go through them. Some of them are just based on legacy. Some of them are based on the fact that we often work with commodities that are just not well understood and not well studied, and there just aren't many tests available. We really don't want to have to go through that rigorous process of developing and validating new tests if we can avoid it. We do that, of course, because we have to. But if good tests are already out there, even if they're 20 years old, sometimes that's the better option. However, in that setting, the main bane of our existence is viruses, or viruses and viroids, but I'll just say viruses, which when I say that, I'm including viroids. So 2/3 of everything we test for are viruses. So it takes up a massive amount of our workload. So I think I kind of mentioned this already, just about the amount of material that comes through the facility. We have to test for over 250 individual viruses, and each individual plant. Some of them can get up to 25 tests. So again, just a lot of work. And ultimately, again, it's just not sustainable. We have to find a better way to doing this, because every time we find a new or emerging virus, or a virus moves into another new country that are sending us material, the list of tests just grows and grows. So we have to change because it takes too long, takes up a lot of space. Some of that testing that I talked about requires horticulturalists to produce plants for us to test on in our glass houses, so that takes up space that we could otherwise use for imports. Labor intensive, and a real problem for us as well is it requires, a lot of this work requires a real traditional plant pathology skill set, which just is getting harder and harder to find. Universities just aren't creating graduates with those classical plant pathology skills anymore, and that's a real problem. So the approach we've taken for this is a small RNA approach. I'll just go through this really quickly. Essentially, what we do is hijack a nice little response of the plants. We're not actually detecting the virus directly. So when the virus infects the plant, it evokes or invokes an immune response in the host, and that response results in the generation of these short little oligos, and then they essentially kind of move around the plant, and when they recognize and bind to the target invading virus sequence, that triggers breakdown of the virus. So what we do is we actually extract and purify and enrich for those short little oligos, and then we deplete for the sort of ribo-RNA background. And the really cool thing about that is it automatically gives us a significant fold in our sensitivity, because the level of these short oligos in the plant that the plant creates is much higher than what would be from the actual virus. And that's really what almost makes this whole concept work. Looking at time, I'll skip through this pretty quickly. This is just a bit of a snapshot of the kinds of testing we do. Of the results of the side-by-side testing that we've done to really just demonstrate with as much rigor as we can that this whole concept wise. So as you can see, over 400 imported plants have already all been tested side by side with this method compared to how we do it presently. And the important point really is this one, that we find 100% agreement. So we think this method, well, we know this method is at least as good, if not better, than how we've done it previously. And that's really important. As scientists, you know, we think this is great. Yep, no worries. But we can't just go to our policy colleagues in Canberra, which is, you know, a Washington equivalent, and say, hey, you know, trust us, this is great. You can hang your hat on this. We've got to show them solid data and lots of it. Now that's kind of the easy bit. That's the sciencey bit. What's been really challenging with this, and it's taken us 9 years from original first project concept to implementation. The really tricky bit is how to deal with the nuance of the kind of detections we get. So we have to make what can be quite unpleasant regulatory decisions based on this data. You know, if some guy has had his material sitting in our facility for a year or a year and a half, and he's paid $10,000, and then we turn around and destroy it, we've got to be super confident on making that decision. So a couple of quick examples. You know, HTS, okay, you get all this great sequence data, you line it up, bang, there's your pathogen. But as a regulator, what do we do if we find a fragment of the virus? Does it matter if that fragment is a benign fragment or if it's a virulence factor? What if the virus is just unknown to science? And that happens, you know, semi-regularly in the plant world. You know, what if the virus is integrated into the host genome? Does that make a difference? All those kinds of things, those complexities, we have to work through and have decisions and positions in place in advance that satisfy both us from a technical perspective and and our policy colleagues in Canberra as well. And that was really challenging. Now, what does that look like in reality? I've just got two very quick examples. So Fragaria, that's strawberries. Before HTS, every single strawberry plant had a minimum of 11 tests just for viruses. Now they have one. Prunus, so Prunus is stone fruit. So think things like cherries, plums, nectarines, peaches, and so on. Pre-HTS, like 15 odd tests. Now there's only 2. So massive dividends for us in terms of efficiency. So less work. And we can now also detect every virus in one test, not just the ones we're targeting. So that's huge. But other bonuses as well. The importers love it because not only are they getting material at the other end that has lower biosecurity risk, because we have a better understanding of what's in it or what's not in it, but it's cheaper. So great, everyone's a winner. All right, a couple of things I'll just finish up on, a little bit of detail. So all of this, of course, we have published along the way to look more than anything, give the comfort and confidence, not only to, like I said, our policy colleagues, but also to clients. So our business is 100% cost recovered. If you send a plant through our facility, you pay the full costs. We, the government, you know, we pay for the capital, if you like. We provide the building, but the service you pay for. So we need to make sure that the people who are going to send their material through our facility have confidence that, you know, we know what we're doing, and this big new change in how we do business will work well. What we've had to do as well, which has been really chunky, is develop from scratch an entirely new cloud-based bioinformatics pipeline, which we've had to build. That's based on something called Galaxy Australia, which is cloud. It's really awesome. It's all freely available, and it's referred to in particularly that third publication there, if anyone's interested. And the other big chunky thing we had to do was actually build our own custom plant virus and viroid database for which we could search against. You know, we all know that NCBI is a fabulous tool. It's great, but it's not perfect. You know, we need better than that for the kind of decisions we're doing. And here's just a little screenshot of the pipeline that we've created. This is kind of your landing page of where you start doing your bioinformatics. And I think that this is my last slide. Acknowledgements, of course. This is a large part of the team that did it, not exclusively. Particular credit goes to Queensland University of Technology, who have been our primary partner through this entire journey and done really amazing work. And also acknowledgements to Agriculture Victoria, who are leading what is the next step of this work. So we haven't finished here. We want to now expand our HTS capabilities to start moving into bacteria and phytoplasma. And AgVic are leading that work. And one of my colleagues, Brendan Ladoni, is one of the other speakers who is from that group. I think that's it from me. Thanks very much. Let's see if I can work out how to end this.
Thank you, Dr. Dinsdale. If you can please join me if you're able to use your reaction button to give a virtual round of applause for Dr. Dinsdale. I really enjoyed that talk. For the sake of time, we're a little bit over. I'm going to ask if you'll paste your comments into the chat box and we'll try to deal with those. Or you can paste a comment into Hugo. I'm going to go ahead and pivot to the next speaker so you can have enough time to get to this. Thank you again, Dr. Dinsdale, very much for that wonderful presentation. Our next speaker is our second speaker in the lineup for Oceania, and it is Professor Radoni from the Agricultural Victoria Unit of Microbial Sciences, Pests and Diseases, where he serves as the director. He leads a large and diverse research group that focuses on this same topic of biosecurity. And for the sake of time, I'm going to stop there and pass to our speaker. Thank you, Dr. Radoni.
Sorry, folks, I was muted there. I was off. Yeah, look, thanks, Eric and Chelsea, for inviting me to present, and thanks to Adrian as well for giving a bit of background in biosecurity. And as Adrian mentioned, we work in similar areas and collaborate together. And so some of the things that I talk about, Adrian has covered really, really well. But what I did want to focus on today was some of the new technologies that we think are really important from a surveillance perspective. So I'll talk about diagnostics, but really that's diagnostics to support surveillance outcomes. So as Adrian said, we're from Australia. We're down in the bottom corner of Victoria, and I work in Agriculture Victoria Research, and we are employed by the state government of Victoria. And we have a number of smart farms around the state, in horticulture in the north, in the central west region of Victoria. We have a grains concentration, and I'm based right down here in the bottom in Melbourne at La Trobe University at the AgriBio Centre. And these pictures just sort of depict where we're coming from a plant perspective. We're incredibly interested in genetic gain and improving germplasm for Australian farmers. We do a lot of work in breeding and phenotyping and understanding pest and pathogen interactions with plants, and we support this through an applied systems approach where we can support our phenotyping observations with metabolomic or sequencing or proteomic type capability. So to really get down to that cellular level and understanding why we observe a phenotype in a certain situation. Adrian went through these, so I don't think I need to say them too much other than to reiterate what Adrian said, and that is biosecurity pressure right across the world is on the increase, and Australia exports 2/3 of its agricultural commodities. So this is incredibly important that we protect our farmers, and there's twofold reason there. One is if we keep things out, it's cheaper to produce, so we're more competitive, but also to maintain market access. And so our science capability around biosecurity is focused on an innovation agenda, and our clear mission is science and technology for productivity and biosecurity outcomes. We really want to, from a biosecurity perspective, we really want to enhance response and management of plant and animal pest and disease. And we often use this invasion curve to give some sort of perspective about when to act and what's the best time to act. And certainly down the bottom here, this y-axis basically represents this investment to manage. And if we invest in preventing, the cost to Australia and to our farmers is a lot lower than if we have to invest in protecting a crop. So we really want to try and prevent. If we have an incursion, we want to try and eradicate. If we can't eradicate, can we contain before it goes endemic and established? Some of the skill development, we definitely work in staff, and I'd like to put in a plug now and at the end that we will be recruiting. So if there's anyone from the US or from this network that's looking for some work in Australia, please keep an eye out. And Chelsea, I might send you a few position descriptions, but also through La Trobe University, we're always looking for masters and PhD candidates that would love to come and work with us on some of our biosecurity outcomes. For context with biosecurity and diagnostics, I try and break it down along that invasion curve around pre-border, border, pre-border, and that's where Adrian and his team do such a great job for Australia. At the border, again, where Adrian is focused on, and then post-border and market access. And this market access, because we export so much, is really, really important for Australia. And with those four areas, when we think about diagnostics and surveillance, try and consider these three terms down the bottom around identification versus detection versus indication. And those three terms can really define what level of energy or diagnostic investigation you need to do to get to that level. So an identification is an absolute identification that this organism you're looking at is identified to genus, species or subspecies level. And that can often take two, three, four different types of properties that we need to determine. Detection is where we might just identify one of those properties and we've got to, we've detected the organism. If we need to identify it, we need to invest more time. And I think the new kid on the block with diagnostics and surveillance is indication, and our environmental DNA technologies and how we used genome sequencing for our COVID pandemic, I'm sure in the US as well, was used as an indication. And the best example of that, and we'll talk about this later, was when we screened sewage treatment plants for the presence of COVID, and that was an indication. If we had a positive in the pond, that was an indication that COVID was present in an area. And so those three levels we need to talk about and think about when we go down a surveillance road. Then we need to consider diagnostic scope and biodiversity in our environment. And so we did some great work globally with the pandemic, but COVID was on one host to humans. In biosecurity, we have hundreds of thousands of pests and pathogens that affect all the species that we deal with in plant and animals. So the challenge for us is diagnosing these pests and diseases, and it's much, much greater. And the more hosts we have, particularly like we do in agricultural plant-based systems, the bigger our diagnostics and surveillance challenges become. And so I sort of depicted that down the bottom here with our hundreds and thousands. So the surveillance diagnostics conundrum that we think about a lot is that traditional method to collect data from the field and do a diagnosis. The traditional way is to follow these 7 steps where we need to collect the sample, package it, get it to the lab. The lab then needs to receive that sample, lodge it into their own system, conduct a test, report the result back to the field. And then often there's a different data system between the laboratory data system and the surveillance system. So there's recording of data, and this takes time. And for us, and I'm sure it's the same in a lot of our countries, time is money. The more expensive something is, the less we tend to do it. So we then thought, righto, what if we could do our diagnosis in the field? So if we just collect the sample, we conduct the test in the field, and then we enter the results directly into our surveillance database. So all of a sudden, we've gone from 7 steps to 3. The assay that we have spent some time on over the last decade is called LAMP. I'd say most of you have heard of LAMP, and the features that really attracted us to LAMP as a diagnostic tool in the field is that the actual enzyme in that assay is thermal stable, and we can use that enzyme at the one temperature, at an elevated temperature of 65 degrees. The other thing about that enzyme, which was incredibly attractive, and Adrian referred to it, is that it can actually tolerate crude extracts of nucleic acid. So we don't need to invest a lot of our time in getting good quality DNA or RNA like we do need to do for PCR and a lot of our high throughput sequencing approaches. So that was a big bonus for us. It's quick, 60 minutes, and it is comparable to what we can do in the lab when we compare that to PCR. So we thought that was potential, and basically there's 3 steps: nucleic acid extraction. We need to validate the assay and do so specificity, and also do sensitivity assays. And we've done this over here is fire blight, an important pathogen in the US and one we don't have in Australia. So we adopted a LAMP assay from the US actually for use in Australia, but we also developed one for this spindle tuberviroid down the bottom. But it was one of our first assays, and where we started to think, gee, the sensitivity of our LAMP is really very good, and we find that our LAMP assay is within 1/10 of our qPCR assays now. To ensure our surveillance teams and our farmers could adopt this technology, we spent a lot of time in training in the field, top right corner, really work with end users about what they find is easy, but then identify how to get this into a very easy step process so that any of us after 2 or 3 hours training could conduct a LAMP assay in the field. One of the ones we've worked on recently is Asian citrus psyllid, and just go into a bit of detail. Asian citrus psyllid is another pest that's present in the US, not present in Australia, and is a vector of citrus greening or hung longbing. So we don't have the psyllid, so we really wanted to develop a LAMP assay. And so in this phylogenetic tree up here, we can see the citrus psyllid and closely related psyllid species. So we tested all these species as well as the citrus psyllid with our sequence and our assay to make sure it's specific. Then we've done our sensitivity assays, and we've looked at our positive G block versus our in-field detection to make sure that it is very, very easy to interpret a positive result, this green peak in the field. So some of the species we've developed LAMP for, I'm a plant virologist by trade, so I was really interested working with our entomologists on LAMP, but it actually has been a really strong tool for our entomologists both in the field and the lab, because the LAMP works on DNA from just a part of the insect, so a leg or an insect, so a leg or a wing. So if we collect a field sample, we only have one specimen or just a part of a specimen, pupae, larvae, then the LAMP assay does that detection. So it has been really incredible tool. And so for spotted wing Drosophila, for Queensland fruit fly, for Phylloxera, Capra beetle, brown marmorated stink bug, and varroa mite. We have developed those LAMP assays in collaboration with our federal department, DAF. And those names there, if you Google those names, you'll come up with our published papers around those assays. So it's been a really valuable tool for us. To HDS, Adrian gave us a bit of a background on that. What is really, really interesting to note about high throughput sequencing is that this red line represents anticipated use of HDS as a diagnostic tool and as a research tool. And the blue line represents 2018. The red line represents 2026. So we're 4 years away from there. We're somewhere in the middle here in the diagnostics, but this is going to continue to grow. And it's incredibly powerful. But down the bottom, definitely we can use HDS as a characterization tool, identification, detection, or indication. It's a pretty simple, you can break it down into 5 or 6 steps. I was going to say it's simple, but it's probably more complex than that. The workflow that we often refer to is that, and I'll talk more about, is we want to be able to take these mixed samples from the environment, extract some DNA, do some amplification, and identify different species. And to do that is basically these 5 steps where we collect our sample, DNA extract, amplify, which is very similar to PCR. So we have those skills. Then we sequence and we do data analytics. What this does require for use HDS as diagnostic is investment in infrastructure, and that is the sequencer and the computer power, but more really significantly, one of the things I like about as a scientist and a researcher, we actually need to compare our sample data to a database, and that database should include trusted sequence, and that's where our traditional entomology, mycology, virology, bacteriology investment is so important to align a trusted identified species with its DNA sequence. And I think this is a challenge for universities and governments for the next 10 to 15 years, is to build these trusted sequence databases. So just some examples quickly. We did characterization of a bacterium. This is 1 PhD. It was a Xanthomonas species, causes bacterial leaf spot in capsicum. Cut to the chase. We identified that one of these species, Gardneria, is not present in Australia based on whole genome sequence. We identified 2 species of this complex that causes disease on tomato. Another species causes the same disease on capsicum and chili. Really important for management decisions in field. Wasn't known until we compared whole genomes. We also identified other endemic Xanthomonas species that can be associated with disease. So just from that characterization of isolates taken from infected fruit across Australia, that's some of the findings we were able to generate. I think this is probably whole genome sequencing. Targeted whole genome sequencing is going to become a very useful tool. This is the method that our governments used to sequence isolates of COVID during the pandemic. Turned out a result in 24 hours, certainly in Australia, and therefore definitely in the USA. What I would like to draw you to is this serial dilution down here, where we have 1 infected seed in 1,000, and essentially we still had 1,500 copies of whole genome sequencing depth. So the sensitivity of whole genome sequencing is incredibly powerful. And also this technique leads us to multiplexing multiple targets into the one assay. We also, with MinION, we've published this in Defreeze et al. 2022. We wanted to take sequencing to the field. And so we were able to go to a sewage farm just outside of Melbourne and detect and sequence avian influenza virus in scats of wild birds around that sewage farm. And again, it's this extraction, amplification, but then sequencing on the MinION. This is a co-investment with the the Australian Department of Defence, and we really wanted to be able to do this sequencing and the analytics independent of the internet, the cloud, etc. So it's completely mobile, no power. So all our assays did not rely on cool chain. To metabar coding quickly, we using these 5 methods, we have invested a lot of time being able to identify different insect species in a trap collected from the field. And the power of this assay, this is just some of the important species. 5 of them are exotic to Australia, and we can give you that result in the one test. And to demonstrate this best, we worked with the cotton industry over a 6-week period where we trapped insects each day, so 42 traps, and we did that metabar coding on those 42 traps, so 42 tests, and we were able to identify over 200 species at a biodiversity level. And the species of interest at a biodiversity level are listed here. And from a management point of view, we identified these 2 red species, which are aphid pests, and then we identified 2 weeks later the emergence of these parasites that have been introduced to the crop to control cabbage aphids. So this biodiversity output from this test was really powerful, but the industry also interested in pests that are of risk or can harm the cotton crop. And above the week 1, 2, 3 is the actual list of pests that we detected that they were interested in. But our assay also told them that these pests were not present in their crop. So really informative data. Again, all from the one test or all from the 42 tests. And from an Australian point of view, we were also able to screen for exotic pests, and we did not detect any. So from the one test, we had 3 key stakeholders that we're delivering the information to. So we see this as a really, really powerful surveillance tool, and our cost is maybe just maybe 60 to 70% more expensive than just doing a PCR assay at the moment. So if we can get that down, all of a sudden industry will be able to use it. COVID tracing, COVID contact tracing was great to watch from a scientist point of view, and we wanted to invest in this. And the COVID, the contact tracing used HES where we sequenced the genome of the virus. And so we again see a role for this in agriculture. So avian influenza, genomic epidemiology. We had an outbreak of the highly pathogenic influenza at incident premises 1. We sequenced the virus as well as at incidence premises 2 and 3, and we're able to show how the virus moved between those premises. The only problem with this great result was it took us 4 weeks and the information was too slow. So we're really trying to get that information quicker for our regulators and incident control managers. We've also done this at a genome level for insects, particularly with fruit flies. So we've used a genome skimming approach where we can identify populations of the one species and how they're connected. And so this genome skimming is incredibly powerful. It requires an understanding of the genome of the pest, but that's very doable. But then we do this SNP analysis to connect populations. And again, it's a tracing facility that we think is going to be incredibly important, both in an incursion, but also of endemic pests. And my last slide is a working group that both Adrian and I have worked on at a national level. High throughput sequencing is an incredibly powerful tool. It's going to be used a lot more, but like most things, it can be used in a bad way. And so we want to establish standards in Australia that our diagnostic labs work to support biosecurity. And it's essentially around 4 themes. It's around skills, technical, platforms to support us, particularly around those databases, and then policy with our, not only within our labs and our organisations, but with our biosecurity regulators. So that's all I really want to say today. I really appreciate the opportunity, and Chelsea And Eric, I'd love to send you some PDs of positions if you know anyone in your networks, in the GARD network, that may be interested in working in Australia. And we are always looking out for PhD candidates who'd love to spend 3 years in Melbourne, one of the best cities in the world, in the top 20, and have some good fun down here with us. So I might just leave it there.
Thank you, Dr. Radonia. Please join with me and use your virtual applause to give a virtual round of applause for our speaker. We have time for just one quick question before we have to jump into our next GARD event. So does anybody have a quick question you'd like to chime in? I have one if no one else wants to. Okay, I don't see any questions in the chat or who, but please feel free to chime in while we address this quick question. I have a quick question for you, Dr. Radoni, which is, have you all combined, sort of the turnaround time, the accuracy of all of these different diagnostic techs, say LAMP, HTS, and PCR, to look at it from a system scale where you can have these kind of screening tests, and then you have, of course, your gold standard and how best to mix and match them?
The scalability is very important. With LAMP, it's likely not going to be used for 100 samples a day. It's actually likely to be quicker to collect a sample, all those samples, and submit it to a lab. But it's actually a very powerful triage tool. So it can actually concentrate your surveillance teams on areas or properties or, host species to actually spend more time in collecting the sample. So as a scalable tool doing hundreds, I'm not sure LAMP's the answer. With high throughput sequencing, we are very keen on the scalability of that method. We know that you need to invest a lot of time in your pipelines. We call it now the black box, where the sequence goes in and out the other end becomes your result. That black box is automatable, and all the steps to get your sample to the sequencer is just like doing a complex PCR. So you can, liquid handlers and robotics can do that. So we're actually investing in that now, so we can do hundreds of samples for HTS. And that scalability is important because it will make it affordable. In Australian dollars, we can do a PCR assay in high volume for about 70 Australian dollars, which is probably something around $50 US. We think right now we can probably do scalable high number of HTS for somewhere around about 120 Australian dollars, so 80 US, which is slightly more expensive, but I think things will get cheaper. The two things we got to concentrate in is the black box and the database. You compare, because that's your specificity. As a virologist, I'm not concerned with the Amplicon approach with HGS for sensitivity. It is incredibly sensitive. One infected seed in 1,000, and we had 1,500 copies of the whole genome of the virus. It is a sensitive method. But you need to invest time in that black box in your database, and that can take years. And we will now start feeding and continually build on our database for insects and fungi and virus and bacteria to improve our interpretation. But I do think HCS is scalable. I think it becomes more affordable because we give multiple results to different people. They may want to co-invest in the one surveillance sample from the field, and that's another game changer. You're on mute, Eric.
Oh, thank you. Thank you. I really appreciate the portfolio approach that you all are using. I think it's really valuable and insightful. We can try to sneak in one more question if anyone has one, and then we need to quickly jump to our next event. Any last minute questions in the Whova, the chat box? Okay, not seeing any, I'll say thank you again very much, Dr. Rodoni. Please, everyone, if you can take a moment and give a virtual applause for our speaker. We really appreciate your time. We had two great talks today, and it was really nice to hear about the biosecurity. I wasn't aware of a lot of these programs. Very interesting.
No
Please do
problems.
send those. Please do send those calls to us. We'd love to distribute.
Excellent. No, look, I'm sure Adrian's still online. He'll say the same. Thank you so much for asking us to present. And if there's any other topics that you think might be of interest from Australia, we might be able to connect you up with other speakers. So yeah, it's great talking to you guys.
Yeah, we would love
Absolutely.
to. We would love to get your advertisement for PhD graduates. I have graduating PhD students in my lab.
Excellent.
Likewise. Likewise.
Fantastic.
We will be in touch. It was really nice to hear about your work, and Dr. Dinsdale as well.
Thanks a lot, folks. Take it easy.
Thanks so much.
See ya.
Take care.